In our previous lecture, we focused on the heart, but today we're going to look at blood vessels. Our blood vessels are tubes that carry blood away from the heart towards our body's tissues and then return the blood back to the heart. Arteries are the ones that carry blood away from the heart, whether it's oxygenated or deoxygenated. Your arteries branch extensively and give rise to arterioles. Arterioles branch as well into capillaries. Capillaries are the sites of gas exchange between blood and the surrounding tissues. Your capillaries merge to form venules. Venules merge to form veins, and veins merge with each other to form progressively larger veins that carry blood back to the heart. Again, whether it's oxygenated or deoxygenated. If the blood vessel is large enough, it's going to have its own set of blood vessels to supply it with oxygen and nutrients and to rid it from wastes. These vessels are called vasa vasorum. Now, the term angiogenesis is used to describe the process by which new blood vessels grow. Now, an example of angiogenesis is whenever we have a malignant tumor, it's going to secrete tumor angiogenic factors that stimulate blood growth in order to nourish that tumor. But angiogenesis is not always a bad thing. The basic structure of our blood vessels is pretty uniform, but there are subtle differences. All of our blood vessels have a lumen through which blood flows. The walls are primarily made up of three major layers. The innermost layer is called the tunica interna, or the tunica intima. It's made up of an endothelium, which is one layer of simple squamous epithelium that is continuous with the endocardium of the heart. It has a basement membrane made up of collagen fibers, and it has an internal elastic lamina, which are thin sheets of elastic fibers that allow for the diffusion of materials. The middle layer is known as the tunica media. It's usually the thickest, but there are exceptions. It's variable in different vessels. Now, it's made up of elastic fibers that provide compliance and smooth muscle fibers that regulate the diameter of the lumen wall. The last layer, or the outer layer, is the tunica externa. It contains nerves, and it's made up of elastic and collagen fibers, as well as less prominent external elastic lamina. Within this layer, if applicable, we're going to find vasa vasorum, which again are small vessels that supply blood to the blood vessel tissues. Now we're going to focus on each of the vessels individually. Arteries, as I mentioned, carry blood away from the heart. They can undergo vasoconstriction and vasodilation. Vasoconstriction is due to vascular smooth muscle tissue contracting in order to decrease the diameter of the lumen. Whenever we have an increase in sympathetic stimulation to the artery, we're going to have vasoconstriction. Vasodilation involves smooth muscle relaxing to increase the diameter of the lumen. Again, it's due to sympathetic stimulation, but in this case, a decrease in sympathetic stimulation. We have two major types of arteries. Elastic or conducting arteries are the largest type. They have elastic fibers, and they recoil during relaxation of the heart to force blood to flow. They act as pressure reservoirs. Examples of elastic arteries include the aorta, the pulmonary trunk, and its branches. The second type are muscular or distributing arteries. They're medium-sized arteries. They have smooth muscle tissue that is capable of greater contraction and dilation to regulate blood flow through our target tissues. They always maintain a partial state of contraction to keep pressure efficient as well as blood flow efficient. Examples of muscular arteries include the femoral arteries, the axillary arteries, the brachial artery, etc., etc. Arterioles are small branches off of an artery that deliver blood to a capillary. Through vasoconstriction and dilation, they play major roles in regulating blood flow and blood pressure. They do so by regulating the resistance to blood flow. Therefore, we call them resistance vessels. Now, a meta-arteriole is the terminal end of the arteriole, and it helps connect an arteriole to a capillary. At its junction, the meta-arteriole becomes a precapillary sphincter, which is a ring of smooth muscle that controls blood flow into the capillary. Capillaries are the U-turns of the circulatory system. They're microscopic vessels that connect an arteriole to a venule. They're involved with microcirculation, since blood cells often have to fold in order to pass in a single file line through their lumen. Their main function is the exchange of nutrients and wastes with the surrounding tissue cells. Therefore, we call them exchange vessels. Their anatomy is perfectly designed for diffusion. They are only made up of two thin layers, specifically the endothelium and a basement membrane. As we previously stated, meta-arterioles are the ones that directly connect to capillaries. They usually supply 10 to 100 capillaries that form a network of capillaries known as a capillary bed. A thoroughfare channel, on the other hand, is a single capillary-like vessel that directly connects an arteriole to a venule. True capillaries, though, usually emerge from arterioles and meta-arterioles and form a capillary bed. They're not associated with a direct flow from arterioles to venules. A portal system is a network of blood vessels that delivers blood via a portal vein from one capillary bed directly into another. I'm sure you've heard of the hepatic portal vein before. It's found in the liver, and it deals with hepatic portal circulation. Deoxygenated blood from the GI system first travels through the liver via the hepatic portal vein, and then it returns to our body circulation. Another portal system is through the pituitary gland, and another one is found in the kidneys. We have three main types of capillaries in the body. The first are known as continuous capillaries. As their name states, the endothelial lining forms a continuous tube. They have tight junctions between their endothelial cells, along with intercellular clefts that allow for the passageway of small molecules such as ions, hormones, water, and glucose. They're most numerous in the body. The second are known as fenestrated capillaries. They have pores between the plasma membranes of their cells, which allow these types of capillaries to be more permeable than continuous capillaries. We find fenestrated capillaries in the kidneys, in the villi of the small intestine, and in endocrine glands. The last type are known as sinusoids. They have a wider diameter than our typical capillaries, and they're more tortuous. The plasma membranes have large pores and intercellular clefts, and occasionally they can have an incomplete or absent basement membrane. They allow for the passage of proteins and even blood cells between the tissue and our bloodstream. We find them in areas such as the liver and the spleen. We also find them in the adrenal glands and the pituitary gland. Many capillaries merge together to form postcapillary venules. These postcapillary venules then come together to form muscular venules. These vessels deliver blood from the capillaries to larger veins. Therefore, we call them afferent vessels. They have thin walls that are porous, so they can still have gas exchange or nutrient exchange between the tissues and the bloodstream. Our body's veins are usually more numerous than our arteries. They have relatively thin walls because their tunica interna and their tunica media are thin layers. Many of our veins have valves that prevent the backflow of blood and help blood move toward the heart. These valves are made up of two or more folds of the tunica interna that form cusps that project into the lumen of the vessel. We have different types of veins. Some are known as vascular or venous sinuses. These vein-type vessels have a thin endothelial wall but lack a smooth muscle layer. Therefore, they cannot alter their diameter. Who can think of an example of a venous sinus? If you said the coronary sinus of the heart, you're correct. Good. Now, we have superficial veins that travel through the hypodermis, and we have deep veins that travel below the fascia between the skeletal muscles. In the upper extremity, we have more superficial veins than deep veins, and in the lower extremity, we have more deep veins than superficial veins. Have you guys heard of varicose veins in the past? Well, varicose veins refer to veins that have leaky valves. Therefore, our veins become dilated and twisted since blood pools and fluid leaks into our tissues. We can have varicose veins in the esophagus and in the anal canal. We call them hemorrhoids. We can also find them superficially in the lower extremities. So how does blood return back to the heart, even though we have gravity working against us? Well, the body has developed three mechanisms. The first is known as the respiratory pump mechanism or the thoracic pump mechanism. It's due to pressure changes that happen in the thoracic and abdominal cavities when we breathe. Specifically, when we take a deep breath in, the thoracic cavity expands and pressure within it decreases. The diaphragm flattens out and moves inferiorly. Therefore, it increases abdominal pressure. Now, as a result, the inferior vena cava found within the abdomen is kind of squeezed, and therefore the blood found in it is propelled in a superior motion towards the right atrium of the heart. The second mechanism is the muscle pump mechanism. Whenever our muscles contract, they compress the vessels found within them. When our muscles relax, they decompress the veins. Therefore, the cyclic contraction and relaxation kind of squeezes blood back towards the heart and overall increases venous return. Now the last mechanism is a simple one. The valves found within our veins prevent the backflow of blood, so that veins pretty much act as a one-way street back towards the heart. Whenever various arteries supply the same body region, we call it an anastomosis. Since it provides alternate routes for blood to reach a specific tissue and organ, we also call an anastomosis collateral circulation. Now, this not only happens with arteries, it can also happen between veins and between arterioles and venules. Some arteries are known as end arteries and do not form an anastomosis. Now, what happens when these end arteries become clogged or when they die is that it interrupts blood flow to that specific tissue or organ, and it may result in necrosis of the tissue. Necrosis is a fancy term for death. Now, at rest, systemic veins and venules pretty much contain 64% of our blood's volume, so we call them blood reservoirs as a whole. When more blood is needed anywhere else in the body, vasoconstriction reduces the volume of blood in venous reservoirs in order to provide a greater volume to the active tissues. An example of this is during exercise. Now, whenever our body undergoes traumatic damage, such as a hemorrhage, our veins compensate for blood pressure changes by vasoconstriction. Most of our blood reservoirs are found within the abdominal organs and in the skin. Therefore, have you ever watched a video or a show where a person is bleeding out unexpectedly and then they check the abdominal region and ta-da, they found the source? Well, this is why. Our blood vessels are organized into parallel circuits that deliver blood to our various organs. We have three main circulatory routes. The first is systemic circulation. Within it, we find all the blood vessels that carry blood from the left ventricle through the body's organs and back to the right atrium. Systemic circulation has three subdivisions. The first is known as coronary or cardiac circulation, and we've talked about it before. It supplies the myocardium of the heart. The second subdivision is cerebral circulation. It supplies the brain with blood. We'll talk about it later, but it's called the Circle of Willis. And lastly, we have hepatic portal circulation, and it's blood flow through the GI tract to the liver and then back to systemic circulation. The second circulatory route is known as pulmonary circulation. It includes all the blood vessels that carry blood from the right ventricle through the lungs and then back to the left atrium of the heart. Lastly, we have fetal circulation. It exists only in the fetus, and it consists of structures that permit exchange of substances between the developing fetus and its mom. Systemic circulation involves the arteries on the left and the veins on the right. They help deliver oxygen and nutrients to the tissues, and they help remove carbon dioxide, wastes, and heat from our tissues. All of our systemic arteries branch off from the aorta. All of our systemic veins deliver blood to the superior vena cava, inferior vena cava, and the coronary sinus. You're not required to know the name of each of these arteries and veins yet, but by the end of the course, we will have talked about, and you should know the location of each of these. Now let's finish up by discussing fetal circulation. As the fetus develops in the womb, his or her lungs and GI organs do not work. They actually start to function after birth. Therefore, the exchange of materials between fetal circulation and maternal circulation is important and occurs through the placenta. Oxygenated blood from the placenta is carried to the fetus via the umbilical vein. Some blood enters the fetal ductus venosus, which is carried then into the inferior vena cava. The rest first enters the liver via the hepatic portal vein, and then returns to systemic circulation via the inferior vena cava. This blood then moves into the right atrium of the heart. In the fetus, there's an opening between the right and left atria called the foramen ovale that allows blood to flow from the right atrium directly into the left atrium. This hole allows us to bypass pulmonary circulation. Remember, the lungs are not developed in the fetus, therefore it's kind of pointless to have blood flow through them. From the left atrium, blood then flows into the left ventricle, through the aorta, and then into the body's tissues. Some of the blood from the right atrium, though, does manage to enter into the right ventricle. It's pumped into the pulmonary trunk, but again, in the fetus, there's a special connection between the pulmonary trunk and the aorta called the ductus arteriosus. It directs most of the blood away from the lungs and back into the aorta. Some of our blood moves through the aorta and into the internal iliac arteries. From there, blood moves into the umbilical arteries, and therefore it re-enters the placenta where carbon dioxide and other wastes from the fetus are taken back up and enter maternal circulation. Functional structures in the fetus become non-functioning structures in the adult. The ductus arteriosus becomes the ligamentum arteriosum. The foramen ovale closes up and becomes the fossa ovalis. The ductus venosus becomes the ligamentum venosum. The umbilical vein becomes the ligamentum teres that hangs off of the falciform ligament of the liver. And then our umbilical arteries become the medial umbilical ligaments. So before we go, let's review the differences between arteries and veins. This is an artery and this is a vein. The thin squiggle is your internal elastic lamina. This tiny layer is your tunica intima. This thicker layer is the tunica media. This layer here with the squigglies again is the external elastic lamina. And then this outer layer here is the tunica externa. As you can tell, the vein is flimsier. We clearly just see the two layers, the tunica media and the outer layer, the tunica externa. Now, the arteries and veins that you're responsible for knowing for this section are as follows. The branches off of the aorta are the ABCs. A stands for arch of the aorta. B is for the brachiocephalic trunk, and it's just this region here. C is for the left common carotid artery, and S is for the left subclavian artery. Now the brachiocephalic trunk divides into the right subclavian artery and the right common carotid artery. The right subclavian artery continues as the right axillary artery in the axillary region, and then continues into the upper limb as the right brachial artery. The left does the same. At the axillary region, it becomes the axillary artery, and then continues as the left brachial artery. Now for the veins. This is your right subclavian vein, and this is your right internal jugular vein. They come together to form the brachiocephalic vein, specifically the right brachiocephalic vein. And yes, it's just this short area right here. The left internal jugular vein and the left subclavian vein drain into the left brachiocephalic vein. Both the right brachiocephalic vein and the left brachiocephalic vein drain into the superior vena cava. This is your pulmonary trunk, left pulmonary arteries. This is the right atrium, the left atrium, right ventricle, left ventricle. In the cadaver, you have your aorta, your brachiocephalic trunk, the right subclavian artery, right common carotid artery, left common carotid artery, left subclavian artery. It's known as the subclavian because it runs deep or inferior to the clavicle, which is right here.